And we're back. Happy Thursday. Today, we're going to go over some work that was published back in 2015, work I did with my graduate student, now Dr. Piyush Sharma, who is a research scientist at a government agency. Now, this work was among the first in doing exploratory analysis, particularly looking at nonlinear methods for LIBS Amino Acid spectra. And at the time, to the best of our understanding, this was the first nonlinear manifold-based method used to analyze laser scattering spectra, specifically LIBS spectra for amino acids. This was supported by the National Science Foundation. They're the two award numbers. And Piyush was my graduate student, PhD he earned in applied mathematics. So the experimental setup with LIBS is as follows. We use laser induced breakdown spectroscopy, and in LIBS, or laser induced breakdown spectroscopy, you pulse a laser on some sample, complex sample, and as you pulse this laser, it absorbs energy and that sample starts to burn. You stop pulsing that laser and you measure the photon energy with a spectrograph, and the spectral lines give you chemical decomposition or the chemical content of that particular compound. And so we had 6 test elements, amino acids. We had water and polysaccharide, or glycol, and 4 non-essential amino acids: aspartic acid, ASP, glutamic acid, GLU, cysteine, CYS, and D-serine, serine D. Now, I'll just refer to them by their abbreviated names out of the interest of time. And so this was our setup, and we wanted to characterize and build a system that would classify these amino acids well from their spectra, from their LIBS spectra, the laser scattering spectra. And so one of the things we wanted to know, some of the goals of this work, what's the impact of the experimental protocols on the pattern phenomena that we see? And so certainly they have a vessel that they put these compounds on before they pulse a laser on them. And so we wanted to know if the experimental protocols were influencing the spectra. We wanted to know how well nonlinear methods identify the pattern phenomena coming from LIBS. We wanted to put together an approach to nonlinear analysis as a way to kind of measure a notion of self-similarity of chemical compounds from their spectra. And then we also want to mechanize the recognition process. How effective are nonlinear methods to do automated categorization, classification of these things? And so this is an example plot from LIBS spectra. And so on the horizontal axis, we have wavelength in nanometers, and these are the spectral returns. And on the vertical axis, we have this spectral intensity in atomic units. And so on this line, we kind of superimpose the different compounds. We have water, D-serine, polysaccharide, cysteine, glutamine, etc. And we superimpose them. And one of the things you see is that for the most part, they're over one another. But in certain wavelengths, you see the spectral lines are a little bit different that distinguish them from one another. And so they're not very different from one another. And so what we started out doing is we took the entire spectrum from 199 nanometers through about 982 nanometers, and we divided it into equal bands 100 nanometers wide. And then we took all of our measurements for our samples, and we plotted histograms of the spectral bands for each of those 8 100 nanometer wide spectral bands. And so this is an example for aspartic acid. And in the next slide, we have an example for glutamic acid. Now, if you look at them, well, they don't look very different. They roughly have peaks in the same location, but there's some minor differences in some of these regions in these 100 nanometer spectral bands from one another. And so we tried another experiment where we took the weighted mean intensities of each one of these 100 nanometer spectral bands. And so we took each spectral line and we weighted it by the probability associated with that 100 nanometer spectral band, and we plotted that weighted mean. So the spectral line times the probability associated with that particular band, and this is what we got. And so we took the weighted mean intensity based on that approach, and if we look at all the compounds along the columns, and we have the spectral band along the rows, we see that for the most part, they're pretty much the same. Except if we look at this band from 599 nanometers to 699, we see that, they're a little bit different. They vary a tiny bit. They're, for all intents and purposes, the same everywhere. We have, say, from 499 to 599, 138, 138, 1.38, roughly 1.38 times 10 to the 5. But if you look at the band 599 to 699, that spectral band, the fourth spectral band, 100 nanometers wide, we have variation: 1.75, 1.6, 1.5, 1.5, 1.3, 1.67. And so we have some bands where we notice that there are some differences, and these differences are very subtle, but other bands, for all intents and purposes, they're roughly the same. And so that means there are certain very subtle differences. Certain parts of your spectrum are responsible for what it means to be one particular compound from another. And this is underlined by highly stochastic nonlinear processes, the jumping of energy levels from one valency level to the next, and then coming back to rest for the valence electrons. So it's a very highly nonlinear process. And so we wanted something that was a little bit more fine-grained than looking at 100 nanometer wide spectral bands. And so we developed a new metric we call entropy density. And what this entropy density is, it's the proportion, a density measure, like a probability density function. But this density looks at the contribution to total entropy for the spectrum contributed by each spectral line at each wavelength. And so we took the log entropy density and we plotted it for each one of our compounds given all of our sample measurements. And so on the horizontal axis, we have the spectral, the wavelength in nanometers. And then along the vertical axis, we have the log entropy density. And so one of the things you'll notice, it's nonlinear. You see that it's noisy. But if you look at the envelope of each one of these entropy density curves for each one of these amino acids plus water and polysaccharide, you'll notice that these envelopes are roughly the same, but there are some minor differences. Let's take D-serine, for example, in the red in the lower left-hand corner. Now, D-serine, you'll notice the envelope of its entropy density plot, it's kind of pointy. It rises faster than polysaccharide, also than aspartame. Now, if you look at the non-essential amino acids, aspartame, cysteine, GLU, they have roughly the same envelope, but D-serine is quite different. Polysaccharide and water are more similar to one another. D-serine is different from everything else, but ASPs, cysteine, glutamine are roughly the same, but you do see some differences in how jagged that envelope is in various parts of the spectrum. They kind of rise. Like, for example, aspartame, if I set my pen here, let's look at cyan. If you look at this region, halfway between 400 and 600, similarly halfway between 400 and 600 for cysteine, halfway between 400 and 600, you notice it's relatively flat for cysteine. For glutamine, it's a little bit bumpy, so it goes up and down. And for aspartame, it really goes up and down. It's more jagged, more V-shaped. And so there are some minor differences in various parts of the spectrum associated, the LIBS spectrum, with each of the essential amino acids, non-essential amino acids, as well as a big difference in D-serine's envelope. So this idea of entropy density versus wavelength is a nice tool that we found to show us and verify for us that there are minor differences in certain regions, and there are certain commonalities between some of the non-essential amino acids to one another. And so we did a dimensionality reduction, PCA, and PCA is a linear method, but we wanted to see what PCA did. to each one of these compounds. And so we did dimensionality reduction, and we found that about 95 or more percent of the variability in the data was explained by the first principal component. So that wasn't necessarily a good thing. And then we plotted this scatter plot, and the scatter plot has the top three principal components, so in 3 dimensions. We plot each one of the compounds from their spectrum that have been dimensionality reduced. And so if you look at it, you see these bands here, and you see D-serine is out here, and then you see glutamine, the amino acid, that's in the blue, in the green. It's kind of mixed in with yellow, which is cysteine, also mixed in with ASP, which is the blue. Right? Those are the ones that were common from the entropy density plot, those envelopes. And so PCA is trying to, not doing a very good job, but you can see some of the commonalities here for those points in this point cloud that are bunched together relative to those that are a little bit separated. You can really see D-serine separated well from the other amino acids. So we then looked to what happens if we use nonlinear methods, specifically manifold methods. And for this class, I won't get into those details since we spent a lot of time talking about different methods. And so we're going to use non-Euclidean measurements, and we're going to use LLE to do the analysis in addition to ISOMAP. Okay, so in ISOMAP, we tried in this plot, we have going from left to right, top to bottom, we have the k-nearest neighborhood, and we needed a minimum neighborhood size of 8 to have a fully connected graph to compute, use Dijkstra's algorithm to compute the nearest neighbor distances to populate our distance matrix D. And if we look as we increase the neighborhood size, we plot the top 3 principal components, project the data onto them, and you'll see what happens as we increase the neighborhood. And so for the smallest neighborhood, k equals 8, it's very local in its neighborhood. And as we increase the neighborhood size, it gets more global in its description. So what you'll see is how this point cloud changes as you go up in the neighborhood. It's getting more and more global, looking at global smoothness or global shape characteristics of the manifold. So here, for the smallest neighborhood, k equals 8, the points are pretty mixed in with one another. K equals 10, we see that D-serine starts to kind of move off in this point cloud. These points of different corresponding to different compounds, they start to separate a little bit, right? So we get a little bit more separation at 15, and then if you look at 30, all the way to 30, well, D-serine is really far off. You see the black is starting to pull away from the center, moving out to one side of this mass, and then you see all the others are starting to kind of push their way outward. Okay. And so these are scatter plots for ISOMAP. We looked at the Scree plots for ISOMAP that explain the residual variance, how much variance is left over as a result of a projection along 1 dimension, 2 dimensions, 3 dimensions as the top 1, top 2, top 3 eigen vectors, principal components, decomposition for this affinity matrix G for ISOMAP. And so we do that. And if you notice here from top to bottom, left to right on each of these graphs, if we look along the horizontal axis, that's the dimensionality. If we look along the vertical axis, that's the residual variance, how much variance is left over. And then along the top to bottom, left to right, each of these graphs is for a different k neighborhood size for ISOMAP. So starting out, k equals 8, we see this dip right around 5 or 4, the elbow of the graph. And then as k increases, you notice the elbow of the graph moves in this direction to the left. And as it moves to the left, that means that a smaller amount of residual variance is explained with a fewer number of principal components, decomposition of this diagonalized affinity matrix. And so here, as k gets really, really big, you'll notice that my residual variance gets even smaller, and with only 2 dimensions, we get a residual variance of 0.002. That's really, really tiny. And so this general trend says that looking at more global shape associated with the geodesic distance among the data points for LIBS spectra, it tends to explain more of the variance with a smaller representation. And so we also tried LLE, locally linear embedding, and with LLE, we again plotted the top 3 principal components, and we looked at the different neighborhood sizes, and well, not the neighborhood sizes we tried k equals 8, k equals 10, k equals 15, and these components were the top 3 features, if you will, that we used in a down project for LLE. And so here we see, again, as we increase the neighborhood size, right, we're looking at a larger neighborhood around each point, constructing its weight, and we are projecting it down, and we're plotting here the top 3 dimensions in that down projection, in that smaller dimensionality. And so you'll see as we increase k, we start to get some separation. K equals 8, well, they're kind of lined up and D serene is different. K equals 10, they're starting to pull apart. K equals 15, they're starting to organize together. So we have mostly blue here, mostly yellow here, mostly green there. K equals 20, now they're starting to really separate. K equals 25, and now K equals 30, you'll notice the blue is starting to wander over to one side, the yellow is wandering over, the green is situated here, and the red is kind of nice and compact on its own. And so we see again with LLE, as you increase the neighborhood size, you're more and more bringing in global smoothness, more global in scope in terms of the shape or the relation to the shape or the curvature of the manifold. And this is just in 3 dimensions. Here's a summary of residual variance, and here we look at just the first principal component and the amount of variance explained for PCA versus classical MDS, which we talked about, and ISOMAP. And so we find for the first principal component, ISOMAP, depending on the k-nearest neighborhood size, gave the highest amount of variability explained in the first principal component. And so this just shows us that this nonlinearity, this respecting of the geodesic relationships between points on the manifold afforded by ISOMAP is actually doing something and is meaningful for LIBS spectro data. And so that just says it's a highly nonlinear process, and so nonlinear methods based on manifold measurements are more appropriate than the linear methods, namely PCA and cMDS. And so here is the Davies-Bouldin Index, and the Davies-Bouldin Index is a measurement of clustering quality. And what the Davies-Bouldin Index represents is the within cluster distances and the across cluster distances, expresses that proportion. And so a smaller here number means, or score, so the DBI score means better. Smaller means that your clusters are very compact, the within cluster distances are small, and they're very well separated. The between cluster distances are very large. And so here we plot the DBI score for PCA and classical MDS. Let me select my pen here. And if we look at this going from top to bottom and left to right, the top row is for PCA, the bottom row is for classical MDS. And for each graph, the number of clusters is along the horizontal axis. So we go cluster of 1 through cluster of 10, and mind you, ground truth, we have 6 different compounds in this experiment. And then the vertical axis is the DBI score, the Davies-Bouldin Index. Smaller means better. And so here, each one of these columns for these graphs represents the number of dimensions or measurement types as resulting from our embedding in smaller dimensional representations. So we have dimensionality of 2, dimensionality of 3, dimensionality of 5, 7, and 10 depicted here. And for each graph, we're listing the DBI score for 1 cluster, 2 clusters, 3 clusters, on through 10 clusters. And what we see here, the DBI score is quite good for 2 clusters for PCA, but as you go up to 5, approach 6 clusters, the DBI score starts to get worse. It gets bigger, right? So smaller is better. And then for PCA, this is the general trend. As you increase the clusters, the DBI score gets bigger. Now, after 6, it might drop a little bit, but not too much. So if we look at cMDS, we notice here, all right, the DBI score starts to climb, and then it starts to decrease a little bit as you increase the number of clusters. Now, let's look at the same graph for our nonlinear methods. In our nonlinear methods, we get a similar behavior. It starts to drop a little bit as our clusters increase after an initial spike. But our DBI score for ISOMAP for K nearest neighbor of 8, K nearest neighbor of 15, neighborhood size of 30. Those are the rows, and the columns are the embedded dimensions. So what we see here, as you increase the neighborhood size, what happens? As you increase the neighborhood size, there's actually a delay, slight delay, in the increase in DBI score as you increase the number of clusters. But once you get closer to 6 clusters, as you go from like 3 or 4 clusters down to up to 5, 6 clusters, we get a decent drop of 0.1 or more in this DBI score. So we do get some improvements afforded by the nonlinear method. And so the natural question is, what happens if you go beyond a neighborhood size of 30? Okay, so let's look at now LLE, the DBI score. Now LLE is doing quite well, and it's doing what we would really want our dimensionality reduction approach to do. So of course, if our neighborhood size is smaller, LLE across the different clustering sizes, when we go from 5 to 6, it shoots up significantly. But nonetheless, even when it shoots up, it's still relatively small relative to the other techniques. And what you'll see here is as you increase the number of embedded dimensions, as you increase the neighborhood size for LLE, that measurement of curvature is more global in its scope. And so you'll see here, especially for K equals 15, as you increase the number of clusters across dimension, you'll notice this nice shape where you start with a DBI score that's high, and then it starts to plummet, and it kind of oscillates up and down a little bit after dropping. You really want that kind of behavior. As you increase the neighborhood size, the DBI score gets better as the number of clusters increases. And this is a nice stable behavior from LLE that for the most part persists as you go across a neighborhood size that's 15 or more. And so this is the DBI summary. And so if we set a small number of clusters and a small number of dimensions of 2 dimensions for the various methods, well, PCA does pretty well, gives you the best DBI score. But that doesn't hold true as you increase the dimensionality to more than just 2, right? Your nonlinearity really starts to improve as you increase your dimensionality. But for a small number of clusters and a small dimensionality, linear does pretty well. Now, certainly for our original unadulterated representation, we had 26,100 spectral lines, and we take the number of atomic units, the spectral energy, as the raw feature. That's unprocessed. Our DBI score in that representation for 2 clusters is 0.28. So yes, PCA is giving you some improvement over raw, but where ISOMAP and LLE really comes into play is if you increase the embedded dimensionality beyond 2. And this held across different neighborhood sizes, 8, 15, and 30, as reported here. And certainly, you can go much higher in the neighborhood size and realize improvements. And so here we have a graph of classification performance. And for this, we use support vector machines for classification. And we ran it with a polynomial kernel of degrees from 1 to 5. And so each one of these graphs here, different colors, represents a different support vector machine. So K1 is degree 1, degree 2, degree 4, degree 5 polynomial. Along the horizontal axis of each graph, we have the dimensionality, so the size of the embedding. So it's 1 principal component on through 10 principal components. We do that for PCA, for cMDS, for ISOMAP, and LLE. We also, along the vertical axis, have the classification accuracy. And so here what we see, as you increase the dimensionality, the number of principal components in PCA, the classification accuracy starts to go up. And it's particularly better when you have a higher degree polynomial for SVM kernel. And that kind of makes sense because a higher degree polynomial can draw nonlinear surfaces to separate your data points for classification, and this is a nonlinear problem. But we do notice that PCA, the best we can do is a degree 5 polynomial kernel, and these error bars represent 1 standard deviation over the 10-fold cross-validation. But you see that we really don't start doing really well with PCA until we get past 6 dimensions. But when we look at cMDS, now remember cMDS uses relative relationships between all the data points, and these data points are over 6 different categories: amino acids plus control group, glycol, and water. And what we see here is that we get about the same behavior. When we go up to 10 dimensions, we hit about 65% classification accuracy, just like PCA. So let's take a look at ISOMAP. ISOMAP uses k nearest neighborhood, and we took a fixed k neighborhood for ISOMAP. It was 8 or 10 size neighborhood, and we use again that polynomial kernel of degree 1 through degree 5. And ISOMAP didn't do very well for a relatively small neighborhood. ISOMAP really needs a better neighborhood. But if we look at LLE, well, look what happens with LLE. LLE returned the best classification performance, superior to everything else. It was well above 70% accuracy, in particular 72% accuracy for LLE. And so this idea of being local and being global afforded by LLE, measuring the curvature of the manifold, seems to benefit classification performance much better. And so here's a summary of the SVM classification. We'll look at the dimensions, we look at the degree of the best performing kernels, and then we look at the neighborhood size. Now for LLE, we decided to let the neighborhoods go and see how big we can get the neighborhoods to try to maximize for our performance. And for LLE, the best performance was turned in with a neighborhood size of 200. That's a big neighborhood, but LIBS is a very highly nonlinear process. And then you'll notice it's easier to tease apart certain compounds from one another when we have a neighborhood that's a certain size. And I'll comment about that towards the end. But for ISOMAP, the best that we got was a degree 4 polynomial kernel with 9 dimensions, turned in a 64% accuracy. Now, certainly this is better than random performance by random guessing of the labels, but it's still not quite as good as LLE. And then you'll notice that PCA and cMDS, they're doing okay with a degree 5 kernel for the SVM. So what that says is that PCA and cMDS's classification performance is really being helped by the nonlinearity of the degree 5 SVM kernel, whereas LLE and ISOMAP do a better job, particularly LLE, with a high-sized neighborhood, 200, is doing a better job in and of itself, and then the SVM kernel is helping it along even further, which is why LLE turns in the best performance. And so what did we learn from this? We were able to find the presence and the influence of microwells influencing the composition of D-serine. The experimental protocol in D-serine was using a material in which this compound resided when you pulse the laser on this compound, and that material was showing up in the spectra. So we were able to identify the influence of these microwells or the experimental protocol on the spectra of D-serine. Now, we also found that pattern phenomena in LIBS are highly nonlinear since LLE with a really big neighborhood gave better classification accuracy than all of the other methods. And what does that mean? It means that the global smoothness is really important when it pertains to these LIBS spectra. And so for ISOMAP, we found in LLE that a good choice of neighborhood size was better in giving you the ability to tease apart certain things. And so the intersample variability between different types of compounds, if you want to tease them apart, you use neighborhood sizes that are in the interval between 8 and 15. So to tease apart different compounds, you use a smaller neighborhood, more local, but to tease apart intrasample the same type of compounds, how they differ from one instance to the next, you need a much larger neighborhood. And this is very key because if you want to characterize something about the experimental protocol for all the D-serines, you use a bigger neighborhood. The global smoothness matters, but for compounds that are different chemical structures, local matters, a smaller neighborhood size. And then we found more generally that PCA and cMDS gave much more widely separated clusters, but LLE gave the highest classification. And so if you're doing clustering for some analysis, bang for the buck, PCA and cMDS are useful, but if you want to do classification, you're better off using something like LLE because that extra mile is afforded by the nonlinear representation. This curvature of the underlying manifold carries value. So Future work that we had planned for this is to find new methods that can give better results for the classification accuracy. And there are some diffusion-based approaches we're considering, Laplacian eigenmaps and things like that, to look at microscale and macroscale phenomena. This idea of self-similarity along the manifold as a way to measure what the entropy density was showing. Basically, take the general envelope and then look at local patches within the general envelope to do that from a manifold perspective. Multi-resolution analyses to look at different scales along the manifold as a way of characterization of both inter and intra-sample variability, and to also add other non-essential amino acids beyond just those that we had in this experiment. And so that's all I had for today. Here are the contact coordinates. Piyush sometimes checks his email, not a lot. He is a working scientist, a research scientist at a government agency, but you can certainly, if you have questions about it, ask him. You can also feel free to send me an email as well. So that's all I had, and with that, we'll end there. And as usual, have a great weekend, stay healthy, stay safe, and I will see you Tuesday.